Good afternoon, everyone. My name is Kurt Malchow, and I coordinate the 2014 Climate College. We are now in the 4th out of 7th lecture of this series, and this class was made possible through the mutual participation of the Scripps Institution of Oceanography, UC Santa Barbara, and the California Department of Fish and Wildlife. Just some reminders before we get started. If you are CDFW staff, please make sure you register for each class for credit. For non-CDFW folks coming in, please do register for automatic reminders for the class. Last class, we introduced some questions to supplement the Q&A session. Last time we didn't have the time to actually discuss this, but at some time we would like to. The intent here is to discuss how department staff can use the information from the course in their work. I will have topics listed here for reference to consider throughout the class. They'll also be up on the Q&A slide during our discussion. And to clarify, this is meant to complement, it's not meant to supplant any questions that you may have for our speakers. We also have two department colleagues, Debbie Hazeltine-Nielsen and Cynthia Catton, to co-moderate the Q&A session for us today. Debbie is a Marine Region Research Partnership Coordinator for the department and also works on the State Wildlife Action Plan, the integration of fisheries and marine protected areas, and climate change issues. Works on the Marine Invertebrate Project. Very pleased to have Debbie and Cynthia with us today, as they also served on the committee to help plan the whole entire course. So it's great to have them here with us today, actively participating. They both bring a lot of marine resource experience to the class. We have additional information on our website as well as updates for each ensuing class. Please check this at regular intervals throughout the course. And for live attendees at this facility, please sign in over at the table on the side and mute your cell phones. Looks like everyone here is familiar with this facility. If not, restrooms are out the door to the immediate left and to the right down the hall. We have as in all of our classes, most of our participants are through WebEx. We are expecting at least 75 to be calling in today. More may be dropping in. Those WebEx users will be muted until the Q&A session, where they can ask any questions to the presenters or our co-moderators through the chat feature. We are continuing in the subject of physical changes to marine systems from climate change. In March, Dr. Nate Mantua gave a presentation focusing on wind, weather, currents, ocean temperatures, and coastal fog. A few weeks ago, these topics were expanded upon to highlight some of the existing and potential effects of these changes on marine ecology, with presentations by Drs. Arthur Miller and Francisco Chavez. This class will focus on climate change effects on hypoxia and ocean acidification. We have two speakers with us today, and since both are presenting through different means, I'm going to introduce each immediately before they give their talk. We'll also be holding the Q&A after both presentations are finished. We're going to start off with Dr. Lisa Levin. Dr. Lisa Levin is Director of the Center for Marine Biodiversity and Conservation and Distinguished Professor at the Scripps Institution of Oceanography in La Jolla, California. Before moving to Scripps in 1992, she was Associate Professor in the Department of Marine, Earth, and Atmospheric Sciences at North Carolina State University in Raleigh. Dr. Levin is a marine ecologist who studies benthic ecosystems in the deep sea and shallow water. Together with her students, Dr. Levin has worked with a broad range of taxa, from microbes and microalgae to invertebrates and fishes. Her recent research has emphasized three major themes. One, the structure, function, and vulnerability of continental margin ecosystems, particularly those subject to oxygen and sulfide stress. Two, wetland biotic interactions as they mediate marsh function, invasion, and restoration. And three, larval ecology of coastal marine populations, with emphasis on connectivity in response to ocean acidification and deoxygenation. Dr. Levin's research has been conducted over the past 3 decades on the margins of the Pacific, Indian, and Atlantic Oceans using ships, submersibles, and remotely operated vehicles to sample and conduct experiments. She has participated in over 35 oceanographic expeditions around the world and served as chief scientist on 12 of these. She is the author or co-author of more than 175 scientific publications. Dr. Levin has served as North American editor of the journal Marine Ecology, as founding editorial board member of the Annual Reviews of Marine Science, as past contributing editor for Limnology and Oceanography and Marine Ecology Progress series, and has edited 5 special volumes on aspects of deep sea biodiversity. Dr. Levin is a Fellow of the Association of AAAS in Biological Sciences, a recently elected Fellow of the American Geophysical Union, and recently served as Pogo Visiting Professor in Namibia and South Africa. In 2011, she was the Anton Bruun Memorial Lecturer at the Intergovernmental Oceanographic Commission, and in 2012 gave the Sverdrup Lecture at the American Geophysical Union meeting. She has participated as steering committee member of the Census of Marine Life Programs on Chemosynthetic Ecosystems and Continental Margins, and the U.S. Ocean Carbon Biochemistry Program, and on working groups for the U.S. National Academy of Sciences, SCORE, SCOPE, NCEAS, and INDEEP. For many years, she served on the San Diego Wetlands Advisory Board, the San Diego Bay Technical Advisory Panel, and as faculty manager of the UCSD Kendall Frost Marsh Reserve. Within UCSD, she has won the Chancellor's Associates Faculty Award for Excellence in Research and has served at various times as representative to the UC Marine Council, Associate Department Chair, Chair of the Committee on Academic Personnel, and Head of the Biological Oceanography Curricular Group. Dr. Levin is presently the inaugural holder of the Elizabeth Hammond and Morgan Dean Oliver Chair in Marine Biodiversity and Conservation Science at Scripps Institution of Oceanography. Please join me in welcoming Dr. Lisa Levin.
Thank you. That introduction was about as long as my talk. Okay, well, it's a pleasure to be here. I don't wear a microphone, right? No, okay. And what I'd like to talk about today, I'm calling ocean deoxygenation. And I guess I can ask the audience right in front of me, how many of you have actually heard that expression before? Anybody? Only one and a half, two people. Right. It's really one of our climate change problems, but it's one that doesn't get a lot of attention. So I'm very pleased to be able to talk about oxygen today. What I'd like to do is just start with some basics. How do we get oxygen into the ocean, and what are the units that we measure it in? I'll talk about the distribution of oxygen in the ocean from a global perspective, looking at oxygen minimum zones. This is a realm I work in a lot, and then talk about how California actually fits into the picture. I'll then discuss biological responses to low oxygen, look at the time and space scales of oxygen variation, referring specifically to the Southern California Bight, where we now do a lot of our work. I'll talk about longer-term trends in oxygen loss called deoxygenation in the East Pacific, and then finally try to close with a discussion of how oxygen stress is actually interacting with a lot of other stressors, and try to convince you that it shouldn't be addressed alone. And we'll talk briefly about some of the possible things that need to be done in the future. So let me ask the question first, where does oxygen come from? We know that some of the oxygen in the ocean is the result of photosynthesis by plants, but a lot of the oxygen also comes from the air and is mixed into the surface waters and then further into the interior simply by turbulence and mixing and wave action and energy. But then we get a lot of oxygen in the interior of the ocean from the process of water sinking at high latitude. Let's see if I can do this with the cursor, maybe. A lot of, I should say in the North Atlantic, this is often the origin of much of the water in the rest of the oceans, and it sinks and comes down through the conveyor belt, and over the course of about 1,000 years, it makes its way, it makes its way into the Indian and Pacific Oceans, and it's very old, and it's been out of contact. So that water along the way loses oxygen, microbes respire, and it's not replenished because that water is not in contact with the rest of the ocean. We also have oxygenated waters. There we go. Oxygenated waters that originate at the Southern Ocean and sink and make their way towards the equator. So we end up with older water in some parts of the Pacific Ocean and in the Indian Ocean. Now, as far as measuring oxygen in the ocean, there's as many different units for measuring oxygen as there are kinds of scientists, and everybody's got their own units, milligrams per liter, milliliters per liter. Sorry, that should be micromolar. I guess that's a PC Mac problem. Physiologists use PO2 or percent saturation, kilopascals. So there's a lot of different ways of measuring that oxygen, a lot of different units, I should say. And I'm probably in this presentation going to be presenting oxygen in the units that they were measured in by the scientists whose work I'm talking about. But I want to talk now, just mention briefly what the definition of hypoxia. Probably most of you have heard that expression, correct? Basically, it refers to the conditions where there's a shortage of oxygen for supporting life. And most people use the definition of 1.42 milliliters per liter or 2 milligrams per liter or 63 micromolar of oxygen as the cutoff. Below that, we have hypoxia. I should say that definition is a little bit arbitrary, and it seems to apply best in shallow water. In deep water, we have defined what we call oxygen minimum zones as being regions where the oxygen falls below 0.5 milliliters per liter, or about 22 micromolar, or 0.7 milligrams per liter. And again, I'll show you with examples that, in fact, that is often a threshold for organisms that have been living in deep water and evolved to cope with low oxygen. So how do we get hypoxia in our oceans? Turns out we get hypoxia the same way, whether it's a natural phenomenon or a man-made phenomenon. And it all starts with the input of nutrients, and this can happen through upwelling naturally, depicted here on a theoretical upwelling margin, or it can occur through agricultural runoff and nutrient input to the ocean through our rivers, and we call that eutrophication when there are excess nutrients. But in both cases, these nutrients stimulate the production of algae, and we get algal blooms that then sink or die, sink, decay, and they're respired by microbes. And those microbes effectively use up the oxygen in the water. And so that is usually the dominant cause of hypoxia, but it alone is often not sufficient to generate what we might call a dead zone or a hypoxic zone. In addition to the algal blooms and intense respiration that comes with them, we usually have hypoxia occurring in waters that are not mixed very well. In other words, oxygen is not brought in from the air and mixed downward, and that can result from stratification of the water or simply from the water being enclosed by an embayment or flushing being prevented by a sill, for example, in a basin. So lack of mixing, plus we often have hypoxia occurring in oxygen-poor source water. So I just showed you how the conveyor belt takes water and carries it through the ocean, and over the course of hundreds and hundreds of years, that's depleted so that we have oxygen-poor source waters in the North Pacific and North Atlantic oceans. Now, probably most of you are familiar with eutrophication. This is the result of intense nutrient input, usually from agriculture, sometimes from other sources. And this is a map of man-made hypoxia, basically eutrophication-induced hypoxia, from paper by Diaz and Rosenberg. And you can see that most of it's focused in the northern hemisphere. I'm going to have a problem here trying to maneuver the pointer without advancing. So we have a lot of hypoxia in the northern hemisphere, especially around centers of population. Now, we also have naturally occurring hypoxia that results from upwelling of deeper waters carrying nutrients, and that tends to occur in the eastern Pacific Ocean, off West Africa, and in the northern Indian Ocean, illustrated here by pink. And as I say down below, we basically get this in regions of upwelling where the water is also old and out of contact from the surface and where there's no mixing or ventilation. Now, what about California, I should tell you that, let me go back a second. The pink areas here are what we call oxygen minimum zones, and if you drop a CTD through one and measure the oxygen profiles, you will see something that looks like this, where the black line here is oxygen. Cursor. Very high at the surface where there's a lot of interaction with the air. There's a little maximum from phytoplankton producing oxygen, but then it drops very, very sharply. This is off Point Loma in Southern California and reaches a minimum here between about 600 and 700 meters. Oxygen's very low, and then it starts up again, and that's because there are better oxygenated currents coming in from below up down here. Now, this profile is fairly typical of an OMZ. It's a little bit deeper off California than in some places, but I want to point out that if you plot pH, it actually looks very similar to the oxygen profile, and that's because the same process that uses up the oxygen, respiration, also releases carbon dioxide, and so more carbon dioxide drops the pH in the water. So oxygen and low pH go together in or co-vary similarly in our deep waters. You can see here a number of oxygen profiles from different parts of the East Pacific Ocean. They all have the same shape, basically, and if you plot pH profiles, you can see that they look more or less the same. Now, where the oxygen minimum zones intercept the continental margins at bathyal depths, you look up here, this happens, oxygen's very low, between probably 100 or 200 meters to 1,000 meters. Where that intercepts, these are midwater features, but they create hypoxic benthos. And this yellow here depicts a ribbon. Sorry. Okay, that's the problem. Thank you. Not being a PC person. So we have a ribbon of yellow here that represents the hypoxic seafloor, in this case along the margin of Mexico and Baja. And we've calculated that over 1.1 million square kilometers of continental margin floor are naturally hypoxic due to this process of upwelling and the creation of natural oxygen minimum zones. So we don't call these dead zones, however, because the animals have had a very long period of time to adapt evolutionarily, and they can cope with those low oxygen levels. Now, the distribution of the oxygen minimum zone, again defined as oxygen less than 0.5 mils per liter or 22 micromolar, it varies with latitude, and you can see that it's very shallow off Mexico and Peru, very thick off Mexico, but it occurs quite a bit deeper off California. Starts around here, and then it shoals again a bit more off Canada. But you can see the thickness varies and the position relative to the surface of the ocean varies. And this is a result of different kinds of oxygenated currents that come into play. There's California. So you can see it's got a thinner oxygen minimum zone, and it doesn't really start until about 500 meters. If we juxtapose man-made hypoxia as a result of eutrophication with naturally occurring hypoxia, that's the black, the man-made's in red, you can see that the distribution of hypoxia along our coastlines is almost opposite. The man-made hypoxia occurs in places that aren't naturally occurring, where we don't have naturally occurring low oxygen. Now, a lot of my research over the last 3 decades or so has focused on low oxygen realms around the world. I've worked on continental margins in the different places you see here, and so what I'm going to do for at least part of this talk is try to give you a sense of what we've learned from all these studies about the effects of low oxygen on ecosystems and organisms. So I mentioned that animals that live in naturally low oxygen areas are present. It's not a dead zone. These animals have quite a few different adaptations to low oxygen conditions. One is that they all have red blood with hemoglobin that has high affinity for oxygen uptake. Another very pervasive trend is that they have a high surface area to volume ratio. So I call these animals sort of frilly and fuzzy because they have amazing arrays of branchial structures. For example, this Branchiana terabellid polychaete's almost as big as the worm, looks just like a lung. We have a lot of gills on the amphinomids. So these animals, even the fish have frilly gills where oxygen's very low. So they enhance oxygen uptake by having more surface area. Some animals do this by simply being small and long and thin so that they're almost all surface. Now, another kind of adaptation that mobile animals have is to move out of the way or to take advantage of low oxygen waters during the daytime when their predators are up about in the surface, and then to migrate up into better oxygenated waters at night to pay back their oxygen debt and also to avoid predators and to feed. So I've got some myctophid fish and krill illustrated here, but there are many, many animals that migrate up and down vertically in the water column and take advantage of the low oxygen zone that's deeper. Now we have So I've been talking about naturally occurring low oxygen. There are places where we have seasonal low oxygen developing or seasonal hypoxia. Certainly off Oregon, since about 2002, they've started having summer hypoxia. But Oregon's not the only place. This occurs off central Chile. It occurs on the West Indian Shelf. And even the Gulf of Mexico, which is our classic dead zone, the hypoxic zone forms, or the low oxygen forms most intensely in summertime when there's a buildup of stratification and less mixing. Once you get a hurricane in there, it more or less goes away. So there are different parts of the world where we see seasonal hypoxia up on the shelf. In the case of Oregon, it's at 20 to 50 meters water depth, so quite shallow. And what is the response of organisms? Now, our best studied areas are actually inshore estuarine or seasonally hypoxic areas, and we know that when hypoxia sets in, animals that are in the sediments will migrate upwards, trying to basically breathe, and they'll build longer tubes, they'll start reaching up into the water. And for animals that are sessile and can't move, if the oxygen gets too low, they'll die. But many animals will actually get up and migrate or move away. And if we look at hypoxia or oxygen tolerances among different taxa, and this is a chart that's been developed for Gulf of Mexico animals, but it seems to apply quite broadly, we find that the thresholds are highest. That is, the animals that are least tolerant to low oxygen are the mobile animals like fish and crustaceans, and shown in red and green here. And the animals that are more tolerant and have lower thresholds for mortality and for exhibiting stress would be the more sessile animals. There's a crab shown here and some infaunal polychaetes. Now, there has been a meta-analysis of hypoxia tolerance done by Vaquier, Sunyer, and Duarte in 2008, and this has sort of become the mantra. It's been very useful to have all the lab experiments that have been published and many of the field experiments compiled, and what you see here is that both looking at lethal and sublethal responses, the fish have the highest thresholds. That is, they're the least tolerant to low oxygen. I should say the fish and crustaceans together. And then bivalves and gastropods and echinoderms have much lower threshold levels for showing sublethal and lethal effects. And so we can sort of look at what levels of oxygen you tend to find effects. In fish and crustaceans, it's between 2 and 4 milligrams per liter. Those are for sublethal effects. You see a lot of lethal effects between 1 and 2 milligrams per liter. And then we have our oxygen minimum zones, where we actually have animals living, including fish, in naturally occurring settings, living below 0.7 milligrams per liter. I think one of the things that brought home to me the fact that this chart mostly summarizes Atlantic species and Atlantic studies is the fact that off California, where we have naturally occurring much more natural low oxygen than is ever found in most of the Atlantic, the animals are much more tolerant. And so if we see what's happening here. If we look at 350 meters, which I was out last week sampling at these depths and also yesterday, we have a huge array of fish that are actually living at 1 milligram per liter, which is somewhere between the lethal and the oxygen minimum zone levels of oxygen. In other words, I think that what we really need to be doing is creating these kinds of meta-analysis and generating the studies for West Coast organisms, fish and invertebrates, so that we have a better understanding of what really generates sublethal effects and lethal effects in the West Coast species. I'm just suggesting that because they live in much more variable and much lower oxygen levels, naturally they've evolved to tolerate these levels. And you can see all these different kinds of fish that are living happily at the 1 milligram per liter of oxygen. However, we do know that when there are low oxygen events, that we can get mass mortality. These events occur off South Africa, for example. I'll show you the Benguela lobster walkout. Basically, all the mobile animals will just walk onto shore. We have mass mortality. In Oregon in 2006, there was a huge pile of dead crabs, and I'm not sure about the cause of maybe some of you will remember the sardine kill in King Harbor. I'm not really sure whether that was hypoxia-induced. One theory was that the fish were driven inshore by low oxygen upwelling events. We also have seen mass mortality off Baja in 2007 at Arandira, which is near Ensenada. There was a combination of factors. There was upwelling, there was an algal bloom, and I think the physical conditions combined with a red tide and led to mass mortalities of animals, of eels, lobsters, fish, crab, and ray. They all washed up on shore. So this is really just to suggest that we can have dead zone type events occurring regionally, even around here, not that far away. There are many other kinds of effects of hypoxia that do not kill animals. We call these sublethal effects, and a lot of those effects can occur on reproduction. So here are some examples. We see reduction in sperm motility and egg maturation at low oxygen. We know that first-feeding fish larvae require higher oxygen levels for survival than the adults in many cases. We've seen, as a result, loss of larval fish habitat. There was a study in the Elbe River over in Europe showing that you can basically lose whole year classes if the larvae don't make it due to hypoxia. There's interannual variation in cod recruitment that has been related to thickness of the hypoxic layer. And closer to home, we have seen blocked migrations of anadromous fish. So if there are hypoxic events or hypoxic water masses sitting along the shoreline, this can prevent the fish from migrating inland and upriver. And we've even seen it for zooplankton migrations, that their vertical migrations can be blocked as well. And so all of these, of course, have reverberating effects, both to the fish stocks but also to food chains as well. Now, I mentioned that mobile animals will get up and move away when you have hypoxia, and they do this, and there's some good documentation for the Gulf of Mexico, where the black zone there shows hypoxia that sets up every summer, or most summers, and that the fish move out of that zone and aggregate around the edges. So this is shown for Atlantic croaker and brown shrimp, and these aggregations are wonderful for fishermen. Basically, it's so much easier to catch the fish when they're all aggregated at those edges. And it turns out we naturally occurring hypoxic areas also have these dense aggregations of animals at their edges. And I just show you some examples. The top here, spider crabs off Oman, and dense fish and shrimp on a seamount, just at the edge of the oxygen minimum zone, where there's just enough oxygen for these animals and lots of food. So in my work, we have studied the macrofauna. These are the small animals retained on a 0.3 millimeter mesh, and all the different oxygen minimum zones you see here we've sampled, and they all show a very similar pattern. Let me pick volcano 7 just because this is easy to reach, with very low densities in the core of the oxygen minimum zone here, shown here in green, the low oxygen zones, and then a peak just a little bit deeper where oxygen picks up. We have these peaks occurring, and then densities fall back down. So we call these high animal densities edge effects. Okay, there they are, pointed out with the red arrows. The other thing that hypoxia does is that it affects different size classes differently. So little animals with high surface areas tend to be less affected than big animals. And this is illustrated on the Pakistan margin. The oxygen profile is shown here on the left, high at the surface, a big core of low oxygen, and then it starts up again. And if you look at the distribution of animals, we find the single-celled animals, the protozoans, love it in the low oxygen zone, but they, too, even have a peak at 700 meters. The metazoan macrofauna, worms and crustaceans and bivalves, they're absent in that low oxygen zone. They pick up at about 800 meters. The mobile megafauna pick up at about 900 meters. They're more or less absent in here, and the same with the nekton. Their threshold is about 1,000 meters. So this creates actually a zonation of different size classes of animals on the margin of Pakistan, with each size class benefiting from the absence of their larger predators. We also get zonation as a result of hypoxia in an ontogenetic sense, and I've got two examples here. This one on the left is from Karen Wishner, and it shows that among the most abundant copepods in the Arabian Sea, Lucicutia grandis, the juveniles are in increasingly deeper waters, or I should say the larval stages. These are copepodite stages, and whereas the adults live at the lowest oxygen levels. So they're zoned by ontogenetic stage, but we see something very similar for Dover sole. These are very old data from Restvetter, but you can see the little sole are shallower in the water where the oxygen is greater, and the adults are down deeper in the middle of the oxygen minimum zone. Same thing with Anapoploma perfimbria, the sablefish. We get small individuals at shallower depths where there's more oxygen, and then the bigger ones live deeper in less oxygenated water. We know that hypoxia or low oxygen can affect food webs, and this is just one figure from a very recent study by Sperling et al. That shows the numbers of carnivorous taxa as a function of oxygen level. Looking for my cursor here, there it is. And that basically when oxygen is between 0 and 0.2 mils per liter, you almost have no carnivorous taxa presence. And as oxygen increases, you get more. And the thought here is that carnivory is a very oxygen-intensive lifestyle. You need to move a lot to catch your prey, and it actually takes a lot of oxygen and respiration and metabolism to digest that prey. And Eric is actually a paleo person who's been able to link this modern-day observation from oxygen minimum zone margins to the Cambrian explosion of body forms, and argues that some of that came from the advent of carnivory related to increase in oxygen in the distant past. Okay. Now, another interesting aspect of food web ecology that's changed by hypoxia has to do with increase in the importance of chemosynthesis. When oxygen gets very low, there are chemosynthetic bacteria that appear, but there are also animals that rely on symbionts. And often those symbionts are using sulfide in the sediments. And so I've got some examples here on top from a gutless oligochaete that lives in a very low oxygen basin off Peru, living off symbionts under its cuticle. We have lucinids in the Santa Barbara Basin, but these pictures here are actually from samples taken on off La Jolla, off Southern California. Point Dume is further north. And we found a lot of animals living via chemosynthesis, and this is something that they're able to do when oxygen is very low. They require a little bit of oxygen, but their symbionts, which are feeding them, often thrive where there is sulfide formed. And I just want to point out that while we think of some of these animals as being associated with hydrothermal vents or methane seeps, a lot of them are actually just occurring in our organic-rich sediments right offshore of California, even without those settings. Now, we're quite interested in knowing what happens when hypoxia or low oxygen affects biodiversity. What are the consequences for different kinds of ecosystem services? And I want to come back to the Pakistan margin, where we've generated a lot of different kinds of data. One, I want to point out to you that if you move through this zone from 800 to 1,000 to 1,100 meters, the diversity changes rapidly. This is a rarefaction curve showing the number of species expected as a function of sample size. And basically, every 50 meters, you get more and more species as you move through. Oxygen picks up just a little bit along the margin. You take an X-radiograph every 50 meters, you go from completely laminated sediments, which are not mixed by animals at all, up here at 700 meters, and you increasingly get more burrows to the point where the sediments are fully bioturbated down here at 1,100 meters. And that has implications for carbon sequestration and the burial of carbon as well. So those animals are part of what we call the biological pump, and they are helping to bury carbon. We've also done some shipboard experiments and in situ experiments that show when oxygen is very low, protozoans are responsible for most of the carbon processing. That would be things like foraminifera, whereas when oxygen is higher, either spatially or temporally, you get animals like worms, polychaete worms, crustaceans, and mollusks being responsible for most of the carbon processing. So there are some fundamental shifts that happen at very low oxygen levels, both in food web structure and in the ecosystem services related to that. I've also done work off India that has taken a look at colonization potential. We put out a series of trays. You can see them over here. 3 different regions on the Indian margin, each with an order of magnitude higher oxygen than the last. And what we found, these were very short-term experiments, unfortunately shorter than I'd hoped, but we did find that colonization is depressed. That is, recovery from disturbance really is depressed at very low oxygen levels, and it can be increases as the oxygen level increases. Let me find my cursor. So no colonization here with very low oxygen, a little bit of colonization when oxygen is 1 to 2 micromolar, and a lot of colonization when it's up to 20. So this just tells us something about the resilience of the community and how well they can recover. I want to talk a little bit about timescales of oxygen variation. The lore is that naturally occurring low oxygen happens on very large time and space scales, and that man-made eutrophication-induced hypoxia, shown here in red, happens on very short time and space scales. But it turns out that's not exactly true. As we start to measure oxygen in shallow water, we are learning more that in upwelling margins, like we have in California, that there's a lot of naturally occurring low oxygen events that occur over short time and space scales. Now, I'm going to give you one example of really long time scales. These are data from the Santa Barbara Basin that show over 1,000-year periods. The basin itself has fluctuated from suboxic to disoxic to oxic, and they know this from the composition of the foraminifera that are preserved in the sediment cores. So those are very long time scales of oxygen change that we have in Southern California, but we have shorter time scales. And on the decadal scale, we have what I call regime scales. We're not sure if these are regime shifts, but this is a plot based on the Calcoffi data of oxygen in mainly Southern California that plots oxygen over time relative to, as an anomaly, that is, relative to the mean. And what you can see is that oxygen was very low back in the 1950s to 60s, went up, got high, relatively high, compared to the mean over the 70s, 80s, and 90s, and then it's been dropping again. and I'll come back to this diagram and how we might interpret it. We don't know exactly if this is a long cycle or not. We do know that there are interannual events that can drop oxygen in our waters. Basically, La Nina can do this, and so here are some data from a paper by Nam et al. That show hypoxic, or at least show that at 35 meters we get a very negative oxygen anomaly over here. Whereas during the that was 2009, 2011, the preceding year we had an El Nino, and that actually reduced production, and so there was less drawdown and less respiration, and oxygen was actually higher than normal. So I illustrate just that we can have interannual variations in our oxygen concentrations. These are all presented as an anomaly, and pH is also plotted over here, along with oxygen, because they vary together in our waters offshore. We also have seasonal variation in oxygen. Again, these are Cal Coffee data in blue, and a continuous sensor off Del Mar at 35 meters in red, and you can see they all show the same patterns. The oxygen gets very low in spring in our coastal shelf waters, and it goes up again in fall and winter. This has implications possibly for the distribution of animals and maybe for management. But we also have wind events that create upwelling that can take oxygen down over just 1 or 2 week periods. So there's a lot to see here, but this chart shows upwelling events in blue and relaxation events in green over a period of time in 2010. And basically we have oxygen going up and down on week-long time scales. Then we also have variation on even much shorter time scales. We have semi-diurnal variation associated with internal tides that on our shelf can take oxygen up and down by quite a few micromolar, and pH does basically the same thing. In blue, this blue section here shows data from 17 meters in the La Jolla kelp forest. You can see lots of up and down. Let's look at oxygen down here. But when upwelling stops and we're in a relaxation regime, things level out, and what shows up is the plant oxygen uptake signal, and that produces a diel signal in pH and in oxygen. So I just want to point out that we have many different time scales of variability of oxygen off California. The same is true spatially. Again, these are data generated 7 and 17 meters in the La Jolla kelp forest. Oxygen is much higher at 7 meters than it is in 17 meters. So animals that are migrating up and down at those depths are going to experience very different oxygen and also different pH levels. Am I running out of time? Okay. In addition to sharp vertical gradients that we have in California waters, we also have inshore-offshore gradients. And so even with a sensor at 3.5 kilometers offshore and one at 1.9 kilometers offshore, we actually see lower oxygen levels inshore in this gray here. Same is true for pH. And if we look at Steve Bograd's compilations of changes in oxygen, we see lots more changes happening inshore than we do offshore. And I want to talk about changing oxygen over time as potentially a climate change problem. We know that when we add CO2 to the atmosphere, we warm the atmosphere, which warms the ocean, which acts to melt ice glaciers and ice caps, and also acts to increase stratification. And when we have greater stratification, we have less vertical mixing in the ocean, and this leads to what we call deoxygenation, the loss of oxygen in the interior of the ocean. It's also important to note that warmer water holds less oxygen, so that also contributes to deoxygenation. And some cases we've had intensified upwelling, and so that low oxygen water is actually upwelled in many cases onto our shelf. So we have a shoaling of midwater oxygen minimum zones, but they are also pH minimum zones. As I said, the same process yields CO2 that reduces the oxygen. And these are coming into conflict potentially with surface-generated ocean acidification, which you'll hear about more from Gretchen. I want to just take a look at, ask the question, has oxygen changed, and how and where has it changed? We'll first start with sort of a synoptic view. Let me just draw your attention to the lower panel here. All these areas in blue are places where dissolved oxygen has declined between the 1960s and the 2000s. And basically, the bold areas are where it's significant. But the message is that all the tropical and subtropical waters are losing oxygen. You can see a lot is also happening in the Eastern Pacific and North Pacific. This corresponds to increased apparent oxygen utilization. But basically, since that time period in the 1960s, we've had at least 4.5 million square kilometers of ocean surface become more hypoxic at 200 meters. So this is a trend, a global trend. It's been well documented in the North Pacific at Station P, where oxygen records go back to 1956. You can see oxygen goes up and down a bit, but the overall trend is for a decline of about 0.67 micromoles per year over time. So the North Pacific is losing its oxygen. And Steve Bograd has also basically summarized CalCoffee data. Since about 25, 30 years ago, he's only looking at this part of the curve I showed you before from about here on down, so oxygen appears to be declining, and the low oxygen hypoxic boundary has been shoaling by almost 100 meters. In other words, the low oxygen conditions are getting shallower and shallower, potentially pushing animals into shallower waters as well. And this effect is most intense right along the California coastline. Something like at 200 to 300 meters off Southern California, we have lost 20 to 30% of our oxygen in the last 22 to 25 years. That's how much the decline is. It's a very dramatic effect. And so what we think may be happening is, for at least some animals, they're probably experiencing habitat compression. So again, we have our migrating fish and krill that move up at night. Well, these same animals are having to move up farther as the oxygen minimum zones are expanding and shoaling. And work by Tony Kozlow has suggested that these animals are becoming much more susceptible to consumption by fishes. And the CalCoffee data show intense decrease in the myctophid biomass over this same time period. And I forgot to mention, part of this habitat compression might be the effect of warming and lowered pH at the surface, which is keeping these animals into a narrower habitable realm and basically compressing their distributions. Another group of animals that this has been documented for are the big billfish, things like sailfish and marlin. It's been shown their distributions are getting shallower and shallower in both the Pacific and the Atlantic as the oxygen declines. And these are huge fish that move fast. They require a lot of oxygen, and it's been suggested that they, too, are becoming much more susceptible to overfishing because of this. What you see below is in that picture is a pursane off Panama, where one big net is catching hundreds of these gigantic fishes. Now, as far as groundfish, they are another group that's quite likely to be affected by hypoxia, and it's been estimated by Whitney and Sinclair that off California, the groundfish that are intolerant of hypoxic oxygen levels are going to lose half their habitat by, this is the present, this is 2050. This has already occurred off Oregon, but it's also predicted to occur off Canada and Alaska. In other words, a shoaling of the habitat simply being pushed up by lowered oxygen. Work by Sam McClatchy has shown for the cowcod already that these species, which live on the islands, Channel Islands that you see here, they lose a substantial fraction of their habitat every spring due to naturally occurring hypoxia. And those are the areas shown in dark, something like 37% of their habitat. But as we continue to have oxygen loss in our coastal waters, the habitable area for cow cod is predicted to decline. And this is just one example, but I think for many of the groundfish, we're going to find their habitable areas diminished by growing hypoxia. Now, not everything dislikes low oxygen. The giant squid, the Humboldt squid, has expanded its range from South America and Mexico, and we now find it up off California and more recently off Alaska and Canada. So as oxygen declines, the squid's done very well. We also have some animals, like these pink sea urchins, they don't show up too well here, that seem to thrive at a pretty low oxygen levels. And so these species may actually be moving up the slope and taking advantage of increased habitat area as the oxygen declines on our margin, and they seem to live perfectly happily at undersaturated omegas and at very low oxygen levels. So the kinds of animals that will experience the changes in oxygen that I'm talking about include those species that attach their eggs or egg capsules to the seafloor, and once they're attached, they can't move, the embryos can't move out of the way. So we see this in market squid. We see a lot of skates and rays, Colletia. There are some fishes, quite a few fishes that do this as well. And these are animals that we probably want to keep our eyes on because and look at where they're depositing their eggs. I know the squid come back to the same habitat year after year, but it's quite likely that those habitats are going to be experiencing continuously lower oxygen levels. In our lab, we have been doing some experiments trying to understand the effects of oxygen and reduced pH together because they co-occur off Southern California. Here's an example with squid egg capsules attached in a bunch of different tanks. They're exposed to what we call sort of normoxic conditions, normal conditions, and then hypoxic conditions are what they might experience during an intense upwelling event. And we have seen that these low oxygen, low pH conditions retard the development of the squid embryos. It doesn't mean they don't develop, it just means it takes them a lot longer, and there are potentially consequences for that. I'm not going to talk about mussel larvae much, except to say that low oxygen and low pH can also inhibit them, and you'll probably hear more from Gretchen about this. But I think it's important to recognize that as oxygen declines on our coastline, and it seems to be, we don't know if this could reverse at some point, but so far it's been declining over the last 25 years, we may be looking at changes in benthic biomass, smaller size organisms, loss of diversity, changes in the vertical range and habitat area of species, and I think we're going to see more microbial carbon processing, maybe more bacterial mats and shorter food chains, and maybe more chemosynthesis happening along at least the outer shelf and upper slope. So there are species that don't seem to do well with hypoxia. I think it's probably not a good idea to generalize across all crustaceans and groundfish, but these are animals whose thresholds and tolerances are relatively high. They're not very good, in other words. They're very susceptible to low oxygen. And then there's other groups that seem to be very tolerant, the bacteria and annelids, nematodes, and some cephalopods and jellyfish and forams, and we may see more of them. So I think one question looking to the future is whether we're going to get alternative types of communities coming in along our margins with different sources of fishery production, potentially different food webs and different carbon processing capabilities. And the last thing I really want to mention is that hypoxia and oxygen loss is occurring in conjunction with other stressors. So we know we have warming, we have ocean acidification happening from the surface, and probably from intensified upwelling below, but then we have organisms stressed by fishing and trawling in some cases, by nutrient influxes and added stressors from eutrophication. And I think it's going to be important as we seek ways to manage that we aren't looking at managing hypoxia alone, but in combination with these other stressors. And then I think a big challenge we have is understanding the consequences of hypoxia, or in this case, I've got pH and dissolved oxygen for all these different processes. And we've studied the effects at a cellular and biochemical level all the way up to the ecosystem level over a variety of ranges of time and space scales. But putting all of this together to try to predict how whole ecosystems are going to respond to all these changes is a tremendous challenge. And of course, it's these emergent properties that we actually most care about. So my closing slide is really a few messages, a few take-home messages. One is that hypoxia is largely a bottom-up effect with consequences for the whole ecosystem, and that we need, where we can, we should be controlling nutrient inputs and sources of other sources of organic matter. I think we need to start paying more attention to sublethal effects, which are manifested at levels that we today don't consider hypoxia. There isn't very much research going on off California in that regard, and I think there needs to be. I think, as I said, hypoxia interacts with warming, acidification, and other kinds of disturbance, and so we have to manage that and water quality together. I think there's a lot to learn from regions with natural hypoxia or that are experiencing other climate perturbations, and I've shown you a lot of examples of these other regions. And I think it's going to be important that we start making continuous oxygen measurements in more parts of our continental shelf and even our estuaries to begin to understand how communities respond and how we can manage in light of declining oxygen. We really need those time series data, and there aren't very many of them. Even the Cal Coffee program's only out there 4 times a year. So with that, I'd like to just acknowledge the fact that all my work on hypoxia over the many years has been aided by a tremendous army of people and various sources of support. And I'll thank you guys.
Dr. Gretchen Hofmann is with the Department of Ecology, Evolution, and Marine Biology with UC Santa Barbara, and she's going to discuss ocean acidification. Gretchen, please take it away.
Hi, everyone. How's that for volume for you all? Okay, Kurt?
Good.
That's good.
Yeah.
Great. Hey, thanks for the invitation. It's really great to be able to talk to you. It's also really nice that we can use these very green methods for communicating, so I'm pretty grateful. So what I wanted to do for you all today was talk to you a little bit about ocean acidification. If you don't know me, I have a lab here at UC Santa Barbara, a bunch of grad students and postdocs, and we sort of do what I would call global change biology and marine ecosystems. We ask, what are the current ocean conditions, how are species currently adapted to this particular environment, and how might species respond to changes in the future. And we do this here in our lab using all sorts of various approaches, but basically we're always doing something in the laboratory to either understand the genetics or to understand physiological processes, and we have collaborators who help us do oceanographic work so that we understand the present-day environment that animals are experiencing right now. So in this presentation for you today that I hope I can name this tune in about 30 minutes, I wanted to just go over ocean acidification as a process, talk a little bit about the impacts on marine life as there have probably been now about 7 to 8 solid years of laboratory and field investigations, and so we can talk a little bit about sort of a, not the greatest detailed view, but the consensus view right now in terms of the biodiversity on the planet. We've only skimmed the surface. I want to touch upon the significance of this, of course, to coastal California, give you a couple examples of projects that we have ongoing in the lab just to illustrate the kinds of biological assessments that people in the community are doing, and then, of course, just really acknowledge that our challenge in all of this, either as oceanographers, biologists, conservation biologists, or our managers, is, we're all sort of trying to predict the consequences of ocean change and link it to what we think the adaptive capacity of these key species are. And I'll come back to that again at the end to talk about how we really are doing that. It's kind of where the rubber hits the road for many of these studies that I'll talk about. So first, just a few comments about ocean acidification. This is an anthropogenic CO2 issue. A lot of people, I think when it first became more visible to policymakers and in the media and in science, it was called, the other CO2 problem, ocean warming's evil twin. And basically, the surface oceans absorbs carbon dioxide, and this creates an acid which lowers the pH and decreases carbonate ion levels in the ocean. And that's really the crux problem here. There are two elements that change in the water chemistry of surface oceans. And I won't delve into this too greatly in terms of scenarios or what's going on, but it's really clear that of the sources of anthropogenic CO2 emissions that we have, the atmosphere holds some, the land and also oceans absorb currently about 27% of the emissions over that time period shown. So people often ask me, Gretchen, is ocean acidification really happening right now? And fortunately or unfortunately, the answer is yes, and we have data that are incoming from locations in the ocean where sensors on buoys have been measuring PCO2 in water. One of the most important ones is at Station Aloha, where in the upper panel in the red and the blue, there's a air PCO2 trend, and the blue sea PCO2 trend shown there. And those show increase in surface water PCO2 over many years, starting about 1989 to 2009. And over this decade, there's been a commensurate drop in pH that correlates with the absorption of anthropogenic CO2 to the surface oceans. So this is a rare occurrence because, as we'll discuss, we really do not have these kinds of long-term data sets coming in coastal regions. But it's very clear that ocean acidification is ongoing and is an environmental problem. Now, the chemistry looks sort of like this. Carbon dioxide hydrolyzes into water. This extra proton is produced, and basically what happens is that it scavenges carbonate ion and reduces the availability of this building block for shells. And so this little graph shows you that as the protons go up, the water becomes more acidic, so that by the year 2000, estimates are the ocean has become about 30% more acidic, and there's been a 16% decline in carbonate ions, those building blocks. And into the future, these are based on, you can find many different scenarios to predict what will happen in surface open ocean. This is what these data are for. There's definitely upwards of 100 to 150% increase in acidity predicted, and with a commensurate drop in carbonate ions. So we understand the open ocean a lot more than we do coastal oceans right now because of the ongoing oceanography, and these predictions apply to those regions. And if you just kind of summarize right now what we know, given the ongoing studies from open oceans, that approximately 28% of carbon dioxide generated by human activities since the mid-1700s has been absorbed by the world's oceans. Ocean acidity, therefore, has increased 30% since the start of the Industrial Revolution. Ocean acidity is actually projected to increase significantly by the year 2100, varying a little bit with different emission scenarios. And the real crux of the matter here is that the current rate of acidification is approximately 10 times faster than any period over the past 50 million years. Geologists have the capacity to look at this and make these calculations, and it's really clear that the rate of change is sort of possibly outstripping the capacity of populations to adapt to changing water chemistry. So the thing that I want to focus us on, given this information about ocean acidification, the place where OA becomes important biologically is that this loss of carbonate ion is deleterious to a group of organisms called calcifiers. These are organisms that form a calcium carbonate shell, and the two most common forms of carbonate materials are aragonite, which is a little more soluble, and calcite, which is less soluble. Basically, one can calculate the saturation state of water. This term is called omega, and at an omega of 1, the precipitation, the formation of a shell, for example, in dissolution, is in equilibrium. And the real concern is that some of the predictions for ocean bodies out there in the future is that this omega value goes below 1, and so many organisms will be challenged to form their shells in future oceans. The other side of this coin is that the altered pH of the water that's driving this change in carbonate ion, it could be that the pH differences in seawater alone are enough to challenge the physiology of organisms. But if we zoom in on calcification, because that's really been where most of the attention has been paid, because it's the obvious connection between anthropogenic acidification and biology. This is data from a meta-analysis done by Christy Croker and colleagues, where they looked at sensitivity to ocean acidification in a variety of calcifiers shown in large groups across the bottom. And then here we're showing effect on size, because it turns out body size is strongly correlated to facing acidification conditions. And interestingly, changes in body size are found in response to many, many different climate change stressors. So this is kind of an emergent property, probably where the physiological costs of performing many maintenance tasks, in this case calcification, comes at the expense of just being big. And so we can see that there's a pretty dominant negative effect across all these different taxa where body size reductions are observed. If the number in parentheses are the number of species, individual species that have been looked at, and that's what I was referring to before. I mean, we have this consensus view from this meta-analysis, but that certainly doesn't even begin to approach the kind of information we would like to have for marine ecosystems in terms of numbers of animals studied. Let's see. If we then also look at different life stages, this is also a very important point, something that I'll focus in on when we talk about the data examples I'm going to show you. It's really clear that there is a life history stage phenomena where larvae tend to be more sensitive than juveniles and adults that have made transitions past key metamorphic stages. And so this is something that a lot of people are interested in, with the idea being that larval forms out in the water column may be very sensitive to these shifting chemistries, and that these are indeed the things that we rely on to maintain and replenish populations. So we'll talk about some actual data from studies like that. So I want to come back a little bit, refocus on the biology a bit. This whole ocean acidification phenomena began with open ocean oceanographers who'd been studying this for quite some time. From an oceanographic perspective, and there were biological oceanographers who sort of put two and two together and realized that there was a critical organism-environment interaction that was going to change, and that was that calcifying organisms or organisms that needed to regulate their pH were going to face these shifting conditions. And so that's opened up this grand challenge of how do we think about an organism, think about all of the things that this organism does in its environment, and try to come up with predictions about how it will do in the future in the face of a changing environment, a changing ocean. And this is really, in a nutshell, global change biology in marine ecosystems. So when we think about this broad landscape of trying to make these predictions and think about populations that we might be managing, it really requires a very big integrated approach across lots of different levels of biological organization, and it involves lots of techniques. So, for example, in this little schematic I've drawn you, we might be looking at the level of the individual, where we might be asking about, oh, well, what is the physiological plasticity of, I think Dr. Levin mentioned squid. So what's the plasticity of today's genotypes to be able to respond to tomorrow's conditions, which is really a great question because, as I mentioned before, environmental change might be going so quickly that, in fact, we have to rely on today's genotypes to tolerate tomorrow's new ocean conditions. So that's one set of studies and questions that we can ask. In the middle here, we have population and multiple populations, a place where genetic variation may play a role, that species adaptation is possible. What would be required is sufficient genetic variation in that population to support the inheritance of ocean acidification resistant traits, if you will. Here's a place where we're asking questions like, wow, is rapid evolution possible? How do we learn about whether or not there are resilient populations out there? And of course, if you're familiar with this area, this isn't unique to ocean acidification research. People think about this with salmon runs up in Canada. Are inland stocks sufficiently genetically divergent and different to be able to tolerate the future warming of inland waters? So this is a very standard view. One of the things that we also think about in this continuum is the biogeographic component, the species range, because we know that there could be species range shifts, and we'll talk about that in a little bit. But one of the things in ocean acidification research is that if there are places where adaptation is occurring, these could be hotspots for adaptation where there are actually resilient genotypes that we might want to think about in a certain way. And if we kind of turn it a little bit, one of the things that a global change biologist will say is that there are about 4 different responses, extinction being one of them, that we can talk about populations making in response to environmental change, and those are geographic range shifts, acclimatization, using that physiological plasticity to counter the conditions that are in the water, or adaptation, true genetic change to respond to environmental change and persist in that range without moving. And that latter one is the tricky one that requires heritable genetic variation. And this is where, in our studies, we're actually thinking that local adaptation is going to matter a great deal. And so when Dr. Levin was saying we need to be looking at California populations very carefully, this is exactly right. And I'm in complete agreement with that from the OA perspective. So that's kind of like a really broad overview. OA, what it means, what it does to animals, the fact that most things that we look at, there's a deleterious impact with a few exceptions. And so I want to kind of bridge to ocean acidification and how might it alter coastal marine ecosystems of California and how we're thinking about this. And I think it's really fair at the outset to say that we know so much less about this physical characteristic of the ocean and how it meets biology, if you will, than we do things like temperature or even deoxygenation, for example, that it's kind of an emerging, ongoing challenge to make our way through these things. But I want to show you kind of where the thinking is, where the field's at, and what we might expect over the next few years. So when I first started studying this years ago now, I thought, I'll just go out and get a little pH sensor because, of course, they make thermometers, and, well, no. The problem with studying carbonate chemistry of the oceans is that there are very few automated or autonomous sensors that you can just bolt out somewhere that'll record pH for you. And so one of the things that happened in the early 2010s, 2010, 2011, is that we collaborated with one of Dr. Levin's colleagues, Dr. Todd Martz at Scripps, and he is a co-innovator of an autonomous sensor that's based on a Honeywell pH electrode. And we were able to deploy these as a community in all these different sites around the world, shown in the map, in several locations in California. And it was really a kind of an influential moment because what we learned, these are just some data from a couple of our study sites. We learned that not all ecosystems behave like the open ocean. If you remember the lines that I showed you for the Aloha site, and that there's also a lot of biology playing a role here that's changing the pH of waters. So on the left are data from a reef in French Polynesia in the island Moorea. In the center, data under the sea ice in Antarctica, and then on the right are some pH data from the Santa Barbara Channel. both those from the island and then some from a kelp forest right near the mainland shown in blue. And right away, you see that there's enormous variation in two sites, with the Antarctic not showing a great deal of variation in pH. And so not all ecosystems are behaving the same. They don't behave like the open ocean. And as a biologist, the really important component here is that it suggests that organisms are probably adapted to and fine-tuned locally, local adaptation. They're fine-tuned to the kind of carbonate chemistry that is prevalent in their local environment. So we started wanting to ask those kinds of questions here on the West Coast, and we've kind of done it with 3 different projects that are color-coded here. So in the spirit of understanding the biology, we needed to get environmental data that we just really did not have a lot of. So we have a group called the Omegas, an NSF-funded group that does research from Oregon down to the south here in Santa Barbara. And this group deploys similar pH sensors and has been collecting pH data in the upwelling region from Oregon down to Santa Barbara. And we're finding that there are differences up and down the coasts, and we're looking at the biology to match that variation in water chemistry. We have sensors in red that belong to the Santa Barbara Channel LTR, and Lydia's sensors, those are sensors that are deployed by a grad student of mine who is funded by the George Melendez Climate Change Fellowship, and she's been collaborating with the National Park and with the NOAA Sanctuary up here to collect data from the field at her sites. So what we were really interested in doing is learning more about the local environment and then doing some of the biology. Just as a note, if we zoom in on the Santa Barbara region, these are the places now where we have intertidal mooring-based or sensors on piers. So, for example, Santa Cruz Island is a place where we have a sensor on the Prisoners Harbor Pier in the national park. So we're collecting data across this region and using this information to parameterize lab experiments and then also to think about what might be happening in the future. And so just to show you what some of these data look like, we've been collecting this for about a year now, and the data are parts of them are available on the SBC LTR webpage, and we'll be having more of those coming out in the next couple months. Here's just a trace showing some of Lydia's data coming from 2 different locations in the Santa Barbara Channel, 2 islands, Santa Cruz Island and Anacapa Island. And you can see the variation on a day-to-day basis, largely due to respiration and photosynthesis activity by the local macrophytes, the kelp. And you can also see patterns that are related, more weekly patterns that are related to, we think, some internal wave characteristics in the area, and some big deflections into low pH zones that are upwelling events, where the pH gets quite low for a period of time when that deep, cold, high PCO2 water comes up on shore. And the fun part about this is sometimes when we're doing experiments in the laboratory, we're bedeviled by this because our intake seawater system in our lab comes right off the coast here, and so all of a sudden our control CO2 levels go way off the mark because our intake has just gone to pH 7.8, and it's hard to sometimes do experiments in the lab. So that's a nice transition to the stuff I want to show you now. So within the ocean acidification community, then, there's this drive to understand the chemistry of the water, the natural variation of the carbonate system for a particular marine ecosystem, and then to know your environment with various sensor deployments, and then to do biological experiments to try to understand the capacities of whatever organism you're interested in to stand future change, or how well do they do if PCO2 doubles in the next 100 years. So there's been an enormous amount of work in that area, and what I wanted to do for you is come back to these early life history stages and just give you 2 examples of how these data sort of come out. The first one is our kelp forest project done in collaboration with the SBC LTR here just off the coast of Santa Barbara. And for this one, a postdoc and a couple of undergrads worked on spores from kelp, Macrocystis pyrifera. Basically, they went out in the field and collected adult sporophytes from our local kelp forest here, and then collected zoospores from these sporophylls that they collected, and then tested how well they would do in the laboratory in the face of variable PCO2. Well, we already knew that the kelp forest, and I think you just saw some data from Dr. Levin, that there's a great deal of pH variation in these coastal environments and in the forest. These are some data, just a snapshot I could find of a data set from Arroyo Quemado, one of our local LTR sites. And so in this particular example, mean pH is about 8.04. The low pH was 7.78. And sometimes, some years and in some upwelling events, it gets as high as 1,500 to 1,800 microatmospheres of CO2. And so we kind of use these numbers to parameterize experiments where after the zoospores were collected, they're shown here like the little spherical thing in the center. These things were held in the laboratory for weeks, and constant water changes trying to look at what would happen to germination of these spores. And basically, in the figure on the right, you've got germination rate on the y-axis, experimental temperature on the x. Both of those are kind of a low and a high exposure for our region of the coast here. And you can see that there was a significant effect of the high PCO treatment, which was really high, but is quite possible naturally and will be more regular in the future. So that here's an example of just a physiological plasticity. Let's ask the kelp spores, how are you doing if PCO2 goes to this level? And the answer is not as well. We also found other negative consequences of more dead spores at high CO2 levels. So it's very difficult. You don't want to extend these kinds of laboratory studies too far out into nature, but it's very clear that the performance, the physiological response of this early stage to elevated PCO2 is not as great as if it was at a low level like 390. So we suspect that this might be going on in nature, and over time, if baseline PCO2 increases and also the spikes of high PCO2 continue to become more frequent in the future, this may have an impact on these early stage spores for this foundational species of the kelp forest. So my next example is also from our shore here, but it's a little more complicated because it gets at that adaptation question. This experiment was done with purple urchins. And in this particular example, we wanted to ask, do different populations that have different exposures to upwelling, like lots of exposure to low pH, are there genotypes that are better, that are more resilient to ocean acidification? And so for this study, we compared a population of urchins down here in Santa Barbara versus a population up north around Bodega Bay from Van Dam State Park, and we knew from our omega sensors that they differed greatly in the amount of low pH water they experienced during the year. And so what we did basically was this fancy cross design experiment where 8 males per site and 15 females per site were crossbred so that northern sires were bred with both northern females and southern females. And what we did with these things is spawned them in the lab. They were reared for 5 days at 13 degrees C. And again, our response variable here was the total larval length. How big is that little larval skeleton shown here as the white glowing image on the right? We had 25 larvae per cross. We measured their body size for a total of like over 3,000 larvae in the whole experiment. And what we found was that, not surprisingly, and we demonstrated this in the past, but that total larval length was lower under high CO2, almost just about 10% smaller. And this is how a physiologist would graph this thing. Yay, here's the average. But population geneticists would do that, so that we break out the percent decrease in size by sire, so that we look here more at the genetic contribution. And what you find is that the sensitivity to elevated PCO2 really varies among and within families. And so if you replot that and look at it a little bit a different way, here you've got columns of Southern California and Northern Californian families. But to cut to the chase here, what really came out of this analysis is that offspring that had a sire from Northern California tended to stay larger. And so the people that did this call this, it matters who your daddy is experiment. And it says that there is genetic variation that confers resistance to OA stress, if you will, on these particular larvae. Now, that's really interesting, but what's even more important is the person who did this, Morgan Kelly, is a population geneticist. And so she calculated a thing called heritability. And for this, the actual number is around 0.5 for high PCO2. And I, not being a population geneticist, didn't fully appreciate what 0.5 meant. So Morgan created this example for me where she said, okay, here's how we think about some traits you'll understand and what their heritability index is. So for Caucasian males in North America, height in Americans is incredibly heritable, and we all know families of people who are super tall, and a lot of the males in the family are not. Now, a group of organisms that I study sometimes, copepods, their size has a really low heritability index. So to have 0.5 is actually quite high for a trait from wild populations like this. So here we are starting to think about natural variation, how to look for genetic variation, and how to think about key populations and use genetics to contribute to the forecast of how things will tolerate future ocean acidification. So this is now, I think, really taken hold in the global change ecology community, and you'll be seeing more of these studies in the future. So to kind of wrap it up a little bit, we're now in this mode of understanding that all marine ecosystems are different, that organisms are probably locally adapted to where they live, be it the variation on a coral reef, where the variation is created by the respiration of the symbiodinium in the coral, to upwelling events on our west coast. And we need to be doing three things actually, getting the physical environment dialed in, but also doing studies that look at these dual aspects of tolerance in animals or algae, that got to look at their different physiologies, and then also look at whether or not genetics plays a component there, because that will also shine light on the persistence of that population into the future. So just a couple concluding remarks. We still have a lot to learn about the capacity of marine organisms to respond to ocean acidification. There are you could go on endlessly with the studies that have been done, but there's some really amazing things. So for example, in calcification in corals, the research community is still kind of not quite understanding the actual cellular mechanisms right down to the surface of calcification in corals that drives that process. And so in reality, when you're studying organisms, there could be this phenomena where, fine, the ocean pH is very acidic, but internally, in the extracellular and intracellular compartments, organisms have really strongly conserved mechanisms to defend that intracellular environment that does calcification. And so that's what Dr. Levin was referring to earlier also, is that these organisms are adapted to a lot more variability and change in their environment than possibly some organisms in more stable environments. That's not to say that that we won't possibly change environments so much that we drive organisms kind of over past their guardrails of tolerance, if you will, that there's probably a limit, a threshold to how much any one organism can tolerate in terms of perturbation of cellular processes. Now, when ocean acidification was first coming to the fore, there was a lot of concern that, wow, everything's toast. And I think Now we have a much more nuanced and complicated environment, but by using all the tools that we have at hand, there is some evidence coming out that does suggest that adaptation may be possible in some species, that evolutionary rescue and rapid evolution is possible. Of course, this is linked to things like generational time, so maybe coccolithophores are going to be better at this than sea urchins that live to be 100 years as adults. So, but these explorations are still really important when we're thinking about managing key ecosystems or key species. And one of the really important points that I can't emphasize enough is that the environmental data to really understand the ocean acidification seascape or the selection regime for the vast majority of at least our California coast region is just missing. A lot of it's technological. We really didn't have the sensors available. More are coming online now, and we hope to have more information about key locations in California soon. And this is globally important. There are groups forming right now. Richard Feely with NOAA has formed something he calls GOAN. I think it's Global Ocean Acidification Network, where people who are recording data from the environment can share their data on this database, and everyone's trying to collect the same metadata, but we're becoming part of that network now. And I think these kinds of efforts into the future are going to be really important. One of the things that's really critical, I think, and it's one of our projects that we're working on in the LTR right now here in Santa Barbara, is that the sensors in the water a lot of times just aren't very sexy, and people are not very excited about them because, it's just monitoring. You're taking observations, and it's just not very, exciting for some people. But the truth of the matter is that getting this kind of information now is really important for us, because there are possibilities that we will identify locations that, for example, have an enormous amount of pH variation, natural variability, and then these places could be thought of as hotspots for adaptation. As I said before, the selection is ongoing, the resilient genotypes are already in those populations, and it might be something we want to conserve. Alternatively, there might be places where we say, wow, there's not a lot of variation in carbonate chemistry there, or we've got seagrass beds that are sucking down carbon like crazy, and it's blue carbon concept. So those are also places that we might want to protect and facilitate. And, one of the consequences for management here, just to, come up with one, is that it could be that our marine protected areas here on the coast of California are also places that we should be thinking about. Are these locations where we can conserve evolutionary potential in the face of coming anthropogenic CO2 changes? So these kinds of monitoring efforts have real value in terms of understanding what the organisms are doing and what they are capable of doing in the future. And lastly, Dr. Levin mentioned this as well, it's really critical that we do not look at these things in isolation, and more people are thinking about ocean acidification, ocean warming, and deoxygenation altogether. Of course, this creates a very challenging research environment, but I think it's one that is becoming ever more cooperative, as witnessed by your climate change college. So with that, I'll wrap it up and open it up to questions.
Great. Thank you very much, Gretchen. Appreciate it. And as we transition, Gretchen, if we can please have you on audio, we're just going to go ahead and transition the screen. We're going to move over to our Q&A discussion segment. Anyone here that's with us live that's got a question, we do have an additional microphone because we are audio recording this. So if you can please just take a seat in front, and then someone who is answering can either use that same one or get behind the podium to add their part of the discussion. That would be great. So with that, one moment, please. Do I have anything from the room?
Well, this is Cynthia Catton. I wanted to thank both of our speakers for actually giving us some insight into what they think we can do as resource managers. I think that they've both given us a really good idea of what we should be expecting from our natural populations out here in the environment off of California, and what we might be expecting in the future, and things to think about moving forward. So I want to take this moment to thank them very much for participating.
You're welcome.
Anything else from the room? Yes.
This is a question for Dr. Levin. You mentioned that warm water holds less oxygen, and I noticed on your slide that showed interannual variability that years of El Nino seem to correspond with higher dissolved oxygen levels. So I was wondering what was going on there.
That's a great question. The effect I showed you, the El Nino effect, is really the result of changes in primary production. And it is true that the waters are warmer up here during El Nino in Southern California, but I don't think the solubility effect is anywhere near as large as the consequences of reduced primary production associated with El Nino. And it's that microbial decay and respiration associated with that production that is diminished when the production is diminished, and so there's less oxygen drawdown. And it just shows how important the biogeochemistry is on our margin and how much of our oxygen condition is related to our primary production.
Thank you.
And this is a question for both presenters. Are there any efforts right now to monitor estuarine, hypoxia, and acidification? Are there any ongoing up and down the coast efforts?
Gretchen, you want to?
Sure. The answer to that is yes. The one that I'm most familiar with, one's in California, one's in Oregon. Professor Tessa Hill at UC Davis, has an ongoing project in Tomales Bay, and so she's been working closely there, not only deploying oceanographic instruments with them, but she's been working closely with the Hog Island oyster farmers and working to think about the productivity of the oyster farm there, and also to look at basic chemistry of the region. And George Waldbusser up at Oregon State at Hatfield Marine Station is kind of an expert in this area, and I know that he's working up north in estuarine region. So we do have these efforts ongoing on the West Coast.
I'm not aware of specific monitoring that's just focusing on hypoxia, but I'm pretty sure in San Francisco Bay that there are some stations that are continuously monitoring oxygen along with many other things. And then most of the NERS sites, the National Estuarine Research Reserves, I'm not sure how many of them have oxygen and pH sensors, but some of them do. Yeah.
Yeah, I think that highlights the problem is that you've got some areas that have, some agencies or groups that have a mission, and they might have one parameter or the other being tracked, but rarely both.
Thank you.
I'm wondering, this is a question for both of you, if you guys could choose additional locations for monitoring both hypoxia and ocean acidification that you guys can't currently get to, where would those places be?
Lisa, you have this one. No, you go for it.
Oh, now I need to think about that one.
Well, I can answer, but
I mean, I think understanding habitat-specific oxygen patterns and pH patterns, carbonate system patterns is going to be important. So we should have sensors in seagrass bed habitats, in salt marshes, in kelp forests, in rocky reefs, in coral reefs. I mean, we need to have no one size fits all. And you all know that the California coastline has many, many different kinds of habitats, and we need to make sure we're monitoring all of them.
Yeah, I mean, that's exactly right, because, the emergent thing that we see is that the biology, or whatever it is, the community assemblage, really hugely influences the seawater chemistry and the level of primary productivity. Just the basic oceanography of a region is a huge driver, and it would be really, I guess, if barring endless time and endless money, it would be really good to sit down and prioritize parts of the coast that are significant to managers and to the state. And so, Tess has done that by looking at Tomales Bay. We're doing that up here, trying to look at inside-outside MPAs to try to just understand what kind of variation we have across those already created management areas. But I'm sure that everybody could come up with, a place that they'd like to have more information about. The list can be endless, but, I think the other thing is to parameterize this with the California economy and just zoom in on what we think is really critical.
One more thing. Yeah, I mean, I think this idea of working from the resources backwards might be very useful, and find, for example, we're monitoring squid egg beds off La Jolla as a place where they always show up. But I mean, finding and different species use different habitats at different life stages. So as Gretchen pointed out, the larval life stages are maybe more vulnerable. So working back from, where do the resources we care about spend their early lives, and thinking about it that way might be useful.
And, that's a great partnership with managers, and, we have a little bit of that going on around here, but, one really important thing is to do exactly what we said. We rally around squid, and we figure out how to study these guys as, a multi-agency group, because we all love squid tacos. And so, and I'm not being facetious, it's one of our most important fisheries. That's kind of why we study sea urchins around here, both the reds and the purples, is because we interface with some of the sea urchin divers here.
Okay, are we both speakers good on that question? We have something in from WebEx, and it reads, for Gretchen, you mentioned that you know more about the role or action of acidification in the open ocean rather than near shore. Why is that? Due to greater variability near shore? If so, then does all that near shore variability help protect some species from the effects of change?
So, going backwards, as a biologist, it may indeed be that organisms on the coast that have all that natural variability are going to be more resilient on average, because there are selected genotypes. We actually kind of find that to be a little bit on the hopeful side. The studies are emerging right now, but organisms in California that are from these upwelling zones do show higher degrees of resilience than those organisms that are from more stable environments where there's not a lot of variation. So yes to the second one. To the first question, I guess I don't want to overstate that we know tons and tons and tons about open ocean acidification. What I was implying is that we're really fortunate that we've had very organized oceanographic data being collected at Aloha out in Hawaii, also at the Bermuda Time Series, and at a station in the Canary Islands. A European group has been collecting data. And because of these oceanographic efforts, we have decades worth of data, long-term data from these sites that tell us a lot about the open ocean. And these are data sets that we completely lack for coastal regions. And, you know, you'll find snippets of data here, snippets of data there, but nothing like what the open ocean sites have. So that's what I was referring to in terms of we know more. That will change in the next 5 years as sensors become more prevalent. And also, you might notice that there's the Wendy Schmidt X Prize, where they're trying to incentivize the innovation community to come up with a really better, more usable sensor so that we can start getting these data very easily on our coasts.
Great, thanks. We have another question from WebEx. It seems like looking at key prey species like squid or others may also be critical. It doesn't specify who's that directed to, but that was the question.
Lisa, take it. It's squid.
What did you say?
Go ahead and take it, because it's squid.
Oh,
Okay.
yeah. I mean, I think that's right. That's what we were just talking about, is trying to identify key resources that the state cares about, in particular for this group, and figuring out where they spend different phases of their life, and working hard to monitor. Basically, if, you know, how we have essential fish habitat that we're trying to identify and define, I think we need to have essential invertebrate habitat for the commercially important vertebrates as well.
Do we have anything else from the room? No questions are coming in. I'm just going to take a quick second just to give a little time in case anything trickles in from WebEx, just to announce that, whoops, I went too far. Our next class is Tuesday, May 13th. Speaker Tom Sukanik from USGS will be discussing sea level change, or rather the inconsistencies along the coast, as well as extreme wind and storm events. That's going to take place back up in Sacramento at our headquarters at the Resources Building. So if we don't have any more questions from both live or WebEx, and I'm not seeing any of those, I want to go ahead and thank everyone, thank the speakers especially, thank everyone and for participating, and we look forward to seeing you at the next class. Thank you very much.
Thank you.